Hi everyone, welcome to Basic Science Series. I'm your host Lokendra Kumar, and today we are going to discuss the structure and the function of an animal cell. On your computer screen, what you're seeing right now is the structure of the animal cell that I have designed using various components that you're seeing on the computer screen on the left and the right side. If you have seen my previous videos, you might have seen that I designed all those components from scratch, and then using those components I try to design a complex organelle like a complete cell, and at the same time I'm going to discuss the functions of individual components. Right now on the computer screen I'm going to design that using those individual components. So it will be a kind of a fun and an interactive way of discussing a specific topic, and at the same time you're going to see how you can model or how you can design a model of an animal cell. Before I start this particular session, I want you to understand that the animal cell structure is quite different from the structure that I'm making right now. It's a schematic representation of the animal cell, but the actual animal cell is significantly different than that. But as you can see by using these diagrammatic illustrations or different structures, at least you will get a basic idea regarding the structure and the function of the animal cell. And also I wanted you to understand that the animal cell, there are different kinds of animal cell. There are significantly modified versions of animal cells, such as nerve cell, and you have cells that are present in our reproductive system as well as in digestive system. There are different kinds of modified or advanced version of the cells. Right now what we're going to discuss is the basic structure of an animal cell, and what are the different components that are present in almost all the cells. At the same time, we are also going to discuss some important facts regarding the animal cell and the structures that are present inside the animal cell. So you're going to have a lot of informative knowledge regarding the structure as well as the function of the animal cell. Alright, so without any delay, let's start designing the animal cell and let's start discussing this structure as well as the function of individual components of the animal cell. Okay, so now you can see that I have removed the structure that I've already designed of an animal cell, and now what I'm going to do is I'm going to utilize these individual components and design that specific structure again. At the same time, I'm going to discuss the function of all those individual components. So let's start by designing the layout using the plasma membrane. Here you can see I have designed this specific structure for the plasma membrane, and we know what is the function of plasma membrane is basically to provide a compartment so that cell can carry out various biochemical reaction. Its important role is also in case of the transportation of the material from outside to inside as well as from inside to the outside environment of the cell. And the structure of this particular plasma membrane is basically composed of phospholipids. And those phospholipids have hydrophobic and hydrophilic ends. And basically this is the basic structure of the plasma membrane, but it is more advanced. If you see the detailed structure of the plasma membrane also contains various proteins, also known as membrane receptors. There are transport proteins that carry out the function of exporting molecules from inside to outside, as I already discussed, and then those receptor molecules, they help in the signaling pathways of the cell. But anyways, we are not going to go into the detailed structure or detailed specific structure of the plasma membrane, but you can see this is the plasma membrane, and it is really important for any cell, right? So next thing I'm going to design is the inside of the plasma membrane, and inside of the plasma membrane is the cytoplasm. And I'm sure you already have maybe in any of your previous classes discussed the function of the cytoplasm. What is cytoplasm? In any cell, cytoplasm is basically the gel-like material that is present inside the cell and the organelles that are present inside the cell except the nucleus, because nucleus has its own nucleoplasm. So right now I can say what I'm making is what I've designed is basically the cytosol of the cell. And when I will add all those individual organelles that you're seeing on the left and the right side, I'm going to constitute the cytoplasm of the cell, right? So you can see right now we have this outline. This outline is known as plasma membrane, and the plasma membrane is made up of phospholipids, right? And the next is the cytosol that I have designed using this specific structure, and right now it's empty. So nothing is in our animal cell that we are going to design. So next thing is the important structure of the cell, that is nucleus. So I'm going to design nucleus. As you can see, I have the basic layout that I've already designed for the nucleus, and you can see these lines inside the nucleus. I will explain these lines. So the nucleus of the cell is basically, it's really important because it contains the DNA, that is deoxyribonucleic acid of the cell, in the form of chromosomes, and chromosome is composed of DNA and the DNA and the histone proteins. Those proteins helps the DNA to pack in the form of chromosomes. Anyways, we will discuss the function as well as the detailed structure of nucleus in our future videos, but right here I'm going to just introduce you about the nucleus that is basically stores the genetic information. And we all know that DNA is composed of nucleotides, and they are arranged in the form of codons, and those codons basically store the information in the form of genes, and those genes, they express specific protein, and on the basis of the specific protein that is being produced, cell performs its function, right? So that is the basic biochemistry of the cell. But anyways, it's more complicated than that. It's not that easy, right? So now I'm going to move another structure that is present inside the nucleus. Is known as nucleolus. Here, this blue colored organelle is present inside the nucleus, and if I'm correct, the function of nucleolus is ribosome biogenesis, and various different kinds of diseases, especially cancer, is associated with nucleus. And a lot of research is going on this specific organelle to understand the process of cancer. So this is present inside the nucleus, and it is known as nucleolus, and you have nucleus which stores the DNA. So as you can see, now we are at this stage. You can see a layout of the cell, you can see the cytoplasm, you can see nucleus, and you can see the nucleoplasm, right? And also the nucleolus. The next organelle that I'm going to design is endoplasmic reticulum. As you can see, I have two different kinds of endoplasmic reticulum. I'll discuss these in detail. Let me just zoom into this specific structure and just align this structure close to the nucleus. This is rough endoplasmic reticulum, and the second structure here, let me also add this particular structure close to the nucleus right here. All right, maybe I need to make it slightly bigger, yeah, to make it as a right fit. Okay, so what you have seen right now, I have introduced two different structures. One is a rough endoplasmic reticulum that you can see is associated with those red dots or brown dots with red outline. And then The second one is the vesicular structure without these red dots. These red dots are the ribosomes, and I'm sure that you know about the ribosomes. Ribosomes basically perform the function of protein synthesis. And rough endoplasmic reticulum has a lot of ribosomes, and smooth endoplasmic reticulum lack these specific structure. And that is the major difference in their structure and the function. If we discuss functional aspect of the rough endoplasmic reticulum is to produce protein in high amount, because it has all the ribosomes, most of the ribosomes of the cell, and that makes it convenient for this particular endoplasmic reticulum, that is rough endoplasmic reticulum, to produce the proteins. And in case of smooth endoplasmic reticulum, that plays an important role in case of lipid biochemistry. Okay, so these are two important structures. Now you can see these are vesicular kind of structure that are present in this inside the cell. Next structure that I'm going to design is this. I'm sure if you have studied biochemistry in any of your previous classes in high school, this particular structure, you have seen this structure before, and this is the mitochondrion of the cell. So mitochondria is basically the structure that is known as the powerhouse of the cell. It generates lot of chemical energy, and that chemical energy is utilized for the proper functioning of the cell, right? And it is also known as powerhouse of the cell. So you need to remember which one is the powerhouse of the cell. It is mitochondria. Sometimes people get confused between endoplasmic, specifically rough endoplasmic reticulum, and Golgi body, as well as the mitochondria function. But the powerhouse of the cell is the mitochondria that generates that chemical energy required to perform the function properly, right? So let me design at least 2 or 3 mitochondria and distribute them throughout the cell. Important fact about the mitochondria is that it contains its own genetic material. It's called mitochondrial DNA. Mitochondrial DNA basically express certain genes that are important for its functioning. So it has its own genome. It has its own genetic information that it utilizes to perform certain functions. So it is really important that you understand there is another set of genes that are present inside the cell, inside mitochondria. And another important fact that I wanted to discuss is regarding the mitochondria is liver cells are the cells that contains highest amount of the mitochondria in animal system, right? And these are double membrane bound organelles. So that you need to remember. All right. Okay. So let's move on to this next structure, and this is peroxisome. Let me just distribute these structures throughout the cell. Peroxisome. If you have seen my previous videos where I'm discussing the plant cell, where I have detailed the functioning of the peroxisomes. Peroxisomes basically produce peroxide, hydrogen peroxide, and that requires to neutralize the free radicals that are generated inside the cell. And free radicals, they are harmful for various functioning, as well as the basic structures inside the cells. They are prone to the damage induced by the free radicals. For example, DNAs are highly susceptible for the free radical damage, so you do not want many like high amount of free radical present inside the cell, and they need to be neutralized, and peroxisome are the organelles that perform that particular function. Now I'm going to introduce another organelle that is, you can see it's a kind of, it looks like a bubble and filled with water, right? Basically it's called vacuole. So we have also discussed this one in case of plant cell, and plant cell has a really large vacuole. It's called central vacuole, and that the function is completely different than the animal cell vacuole. In this case, animal cell vacuole performs the functions of basically storage of minerals, salt, and other important chemical compounds that are important for the proper functioning of the cell. Okay, so right now we have introduced all the structures that are present on the left side, which is cell membrane, which is the outer layer of the animal cell. Then you have cytoplasm, basically cytosol plus all the organelles that are present inside the cell constitute the cytoplasm of the cell. Then you have nucleus that contains the genetic material in the form of chromosomes in case of animal cell. Then you have nucleolus. Major function of nucleolus is the ribosome biogenesis. Then you have endoplasmic reticulum, smooth and rough, mitochondria, peroxisome, and vacuole. Now let's move on to the right side. Right side we have the structure that is ribosomes. So I have these in the form of dots. But basically, the structure of ribosome is more complex than that, and its function is also more complex than you think. So basically, the major function of the ribosome is to produce protein. The major function is to take the information that is present in the genome and translate that information and synthesize proteins. Okay. All right. So that was the basic function of the ribosomes. Now we'll move on to the next organelle, that is Golgi apparatus. And you can see it's a vesicular as well as there are some circular structure that are present on one side. I've tried to design these structures with slightly different color. And these are known as Golgi vesicle, and this particular structure is known as Golgi apparatus. And its main function is basically to transport the proteins that are present inside the cell, and they need to be transported for some reason outside the cell. So Golgi apparatus does the packing of those proteins and then transportation of those proteins using those using those Golgi vesicles, right? So let me introduce these structures. I'll just distribute Golgi apparatus maybe right here. Okay, that looks good. So we have two Golgi apparatus with those small vesicles. You can see that I've already discussed that the main function is to pack those proteins and then transport those proteins outside the cell. All right. What is the next structure? Next structure is lysosome. So let me zoom in this. I'm trying to show you what is present inside the lysosomes. Lysosomes basically are membrane bound organelles, and their function is to degrade unwanted molecules. So what happens inside the cell is a very complicated biochemistry is going on, where the cell is generating a lot of different kinds of material, and some of the material is unwanted inside the cell. So there should be a mechanism to destroy those particular components that are not required in the cell. So lysosomes functions specifically for destroying those particular components, right? So basically its function is to digest those specific components, those proteins, and that is a really important function because you don't want so many unwanted protein inside the cell. So basically the function of the lysosome is to digest the unwanted material present inside the cell, right? And then you have two different structures, which is microfilaments and microtubules. Microfilaments and microtubules, basically they constitute the cytoskeleton of the cell, and they are really important when cell is trying to basically divide. As we know that microtubules, they constitute the centriole, and that is really important in case of the cell division, right? So let me distribute these structures in the animal cell that I'm trying to construct here. And then I'm going to use this particular structure here, or maybe make it a little smaller, and distribute it maybe somewhere here, just to represent that these are the structures that are present inside the cell. But basically what you can do, you can distribute them evenly throughout the cell and make a really nice model of a specific animal cell. With this particular information, what you can also do is you can make a model of the animal cell. You can make these individual components with different things that you want to use. You can use your creative input in that, right? And then constitute this particular model, and this model is going to be really nice looking kind of model. And you can also discuss this particular structure and individual components basically and their function. So this is basically the layout and the model of the animal cell, right? So let me summarize what we have discussed. Basically, in this video, we try to understand what is the structure of the animal cell. We also try to understand what are the different functions of the animal cell. And we have discussed these individual components, right? And if I try to differentiate between animal and the plant cell, there are many differences, but there are similarities also. For example, ribosomes, mitochondria, you have nucleus, nucleolus, cytoplasm, and so many things. They are quite common that are present in case of the plant cell as well as in case of the animal cell. And the most important point I want to make at this particular point is in plant cell, you have a very rigid cell wall. And that is really important for the plant cell. And the cell wall is also present in case of bacteria. And in case of bacteria, you have gram-negative as well as gram-positive cell wall. So you need to understand the differences between prokaryotic and eukaryotic, as well as you need to understand the differences between animal and the plant cell. It's really important. And you also need to focus specifically on these organelles and try to understand what are these organelles in detail and what are the different functions. So in this video, I'm just trying to cover the basic structure of the animal cell, but I will cover the detailed and molecular mechanism of the functioning of the plant, animal, and microbial cell in my future videos. But I wanted to first make videos on those basic topics so that you can understand the way I try to explain things, the way I try to design things, so that it will be easier for you to understand my advanced lectures. For example, I'm going to make videos on cellular signaling systems, where I'm going to discuss some of the immune system signaling. I have already made many videos related to those particular topics, but I wanted to make additional lecture kind of videos so that you can understand the full concept in detail, right? So I hope that this particular video lecture was helpful for you to understand the basic structure of the animal cell. I want you to study individual components. Now, since you are already familiar with the basic structure of the animal cell, I want you to read about these different organelles that I try to discuss and see what you can get, what additional information that you can get from your reading, right? So this is really important. You need to pay attention to the details, because in exams, most of the time, those are the questions that are being asked that basically students are not going to pay attention to, like small organelles. For example, peroxisome, what is the function of the peroxisomes, right? We most of the time focus on nucleus and endoplasmic reticulum, but they are important at the same time. The organelles that are present and you think that is not important, but most of the time questions they are being asked about these organelles. So I want you to read in detail about the cells and also try to remember the differences between plant and animal cell. You can watch my previous videos. I've made videos on bacterial cell, plant cell, and animal cell. You can watch 3 videos and then see what are the differences and what are the similarities among all those cells, right? So with that note, I will conclude this particular lecture, and I hope this video was helpful for you to understand. In future, I will bring more lectures and more advanced lectures that will cover different topics in the area of biosciences. And before I conclude this presentation, I want to thank all my subscribers for supporting the channel and watching the videos, posting your comments on the videos. They are really motivating and they are really helpful, and I hope that in future you will watch those videos and you will also share those videos with your friends and with the other students. And with that note, I want to just stop the video here, and I will see you in the next video. Thank you and take care.